Hi everyone, I'm Adrian and I'm currently a junior at Prospect High under ASDRP in the Department of Chemistry, and today I'll be discussing combinatorial chemistry and its role in enabling both rapid preparation of chemical libraries of analogs and gaining mechanistic insight into medicinal chemistry. So for a quick outline of my presentation, I'll first be talking about the significance of combinatorial chemistry, and then I'll be moving into one of my projects, which involves chemical neuroscience and comparing physostigmine, which is a naturally occurring neuroactive alkaloid, and rivastigmine, its synthetic counterpart. And then I'll talk about our work in synthesizing analogs of rivastigmine. Then I'll be talking about another one of my projects and the biological significance of natural and LSOM, and how we can use this to look into mechanistic studies of the Biginelli multi-component reaction, and finally how we can use Hammett linear free energy relationships to draw conclusions about rate limiting steps. Okay, so with that, before I begin talking about my projects, I want to talk about three themes that revolve around my work. At first, these might seem a little disjointed, but in fact, they work together in quite a unique way to reimagine the way we think about chemistry. So these are combinatorial synthesis, physical organic chemistry, and drug discovery or medicinal chemistry. So combinatorial chemistry enables physiorg studies in rapid time. If you generate one single catalytic platform that is optimized, you can probe mechanism and resulting reaction rates using, for example, linear free energy relationships, as I'll talk about later. And this kind of data is really important when you're trying to improve upon existing therapeutics in medicinal chemistry. So now, now that I've discussed that, I want to talk about a philosophy that synthetic chemists use and when they're trying to make molecules, and that originates from the Industrial Revolution and this idea of interchangeable parts. And interchangeable parts are individual parts of a machine or other entity that are all produced identically. And so then when you assemble this end product, there'll also be like copies of each other's. And this kind of production revolutionized manufacturing because it allowed for easy modification and replacement. And it also dramatically lowered the costs and time and resources spent on production. So over here, I have Eli Whitney, who was an American inventor during the era of the Industrial Revolution. And he's known for producing the cotton gin. But something he also introduced was a manufacturing method based on interchangeable parts. And at first he used this method on manufacturing muskets. However, the benefits of interchangeable parts, because of the ability to diversify, customize, and lower resources required to produce, this kind of ideology was applied to a lot of different things. So during the Industrial Revolution, we see how this evolved in factory production and assembly. And one major example is Henry Ford's Model T, which I think everyone is familiar with. And so we can even see later in history, in modern day, interchangeable parts are even seen when we're talking about pizza. So the toppings in this scenario are the interchangeable parts. Can even apply this to technology like iPhones. And so now we have this idea that we can take a single optimized platform and immediately diversify them however we like. And now chemists can also apply this when it comes to natural product synthesis. And this can be streamlined by taking advantage of our knowledge of molecular symmetry. So chemists are always taking inspiration from nature, especially when it comes to interchangeable parts. In nature, all molecular function depends on the same 4 nucleotides that form DNA, as well as the same 20 amino acids are combined in different ways, shapes, and forms to produce thousands of combinations of proteins and genes that are present in the human genome. For example, there's an estimated 20 to 25,000 protein coding genes in the human genome alone. And this is all deducible to the same simplistic format. And so using nature, we use combinatorial chemistry, and it has major advantages in resolving some of the challenges that that chemists face, and that's difficulty in like discovery and process. So to make a molecule is, especially one that has a very complex synthetic procedure, it takes a lot of time and resources. And if you're trying to make a bunch of different molecules or analogs simultaneously, it's really inefficient to do like a 10-step synthesis, each of which maybe takes like a month of rigorous procedure, when you could just optimize a single synthetic step, and that would be more ideal if you're trying to make all of these at once. So in this case, we have this hypothetical molecule, right? And it can be produced from any combination of these parts, any of these shapes, any of these colors, right? And through a single unified chemical process, you can mix and match to generate a diversity of products that are similar but also different in their respective ways. And now, inspired by the way that nature makes molecules, chemists can now develop ways that, to produce their own reaction platforms and generate a mix and match of different chemical structures that can then be screened for biological activity, and then we can compare these when applied to medicinal chemistry. So in carrying out combinatorial chemistry, we design what are called analogs, and these are compounds with similar structures to the original compound, except they differ with respect to different substituent, for example, that alters its function and the interactions with target molecules. And we can design analogs in a variety of ways. For example, we can exchange functional groups on parts of a molecule. So I'll discuss what this is later, but you can see how we're basically exchanging this functional group here on the left. And we can also create stereoisomers, or the RS and enantiomer, as we do with another compound that I'll talk about later. And in combinatorial chemistry, in which we simultaneously screen a library of these related compounds and for their bioactivity, we hope to determine a product that is optimally suited for a therapeutic or other medicinal function. And in modifying and optimizing a lead compound, we can see if these substituents make our product more or less chemically and biologically favorable. Finally, we can minimize any drawbacks of a compound's particular physical and chemical properties to make it a superior therapeutic agent. So today I'll be discussing how we can use this ideology both in making libraries of analogs in the example of rivastigmine, and use this for mechanistic studies in the example of monasterol. So first, I'll be talking about our work with rivastigmine and other neuroactive small molecules. And you might be wondering why we're so focused on making neuroactive small molecules. The thing is, small molecules are able to pass through the really selective blood-brain barrier in the body, while the movement of larger molecules is blocked. And this makes neurological diseases very difficult to treat because a lot of antibodies that are made can't cross this barrier. And so this makes neuroactive small molecules very important in medicinal chemistry, especially when applied to diseases like Alzheimer's. To show how this works, we have, for example, acetylcholine, which is a neurotransmitter that plays a key role in body movement, memory, and cognition. And when this neurotransmitter is inhibited, this can contribute to disorders such as Parkinson's and Alzheimer's disease. And so we see here a synaptic gap, and after acetylcholine is released, it'll bind to these postsynaptic receptors, and then from there, it can induce muscle contraction, and then is afterwards broken down by the enzyme acetylcholinesterase, which is shown on the right here. So using acetylcholine as a reference, we have physostigmine, which is a competitive mimic of this molecule. And physostigmine is derived from this bean plant called Physostigma venenosum, and it is a dual competitive inhibitor of both acetylcholinesterase and butyrylcholinesterase. So acetylcholine over here is the naturally occurring substrate of this enzyme. Let me get, yeah, there you go. So it's the naturally occurring substrate of this enzyme, and it relies on its cationic site, which has a net positive charge here, and its hydrolyzable carbonyl over here for molecular recognition of this enzyme, and then it can bind to the active site. And this serine 200 hydroxy over here helps carry out this phenomenon. So here we see the cationic site being attracted to the anionic site, and then the active site here. And then in comparison, physostigmine, which is a neuroactive natural product, also competitively inhibits the same enzyme. And it shares this pharmacophore template where it's protonated, and here is the cationic site, and as well as its hydrolyzable carbonate right here. But despite the increased capabilities of physostigmine, it has very low bioavailability. Additionally, it's very toxic and very hard to synthesize. And so, on the contrary, we have rivastigmine, which is an FDA-approved drug that is structurally analogous to physostigmine, but has higher tolerability and bioavailability. And so you can see that it binds to the enzyme in a similar way. And so now we as a group wanted to optimize conditions for synthesizing rivastigmine, which in the past is used sodium hydroxide, unfortunately. And so we wanted to do this without the use of sodium hydroxide. And so we took this table of conditions that we experimented with through lots of trial and error. And then eventually we found a protocol that has optimized yield. Over here, you can see the 57.3% yield compared to basically no yield on here. And so we use DCM, DMAP, and pyridine in here. So now, if you guys remember, I was talking about how we can rapidly diversify with a single optimized reaction platform. And so this becomes our optimized reaction platform. And now we can try to create analogs of rivastigmine and compare their bioactivity. So we were able to successfully synthesize 3 of our intended analogs: diethyl, morphine, and dimethyl, all from our starting material, which is 3,1-dimethylaminoethylphenol, and using different carbonyl chlorides. Of them, diethyl was found to have the lowest binding affinity after physostigmine, while dimethyl was found to have the most binding affinity. We also attempted to synthesize many other analogs that we did not include in our eventual manuscript, and we'll try to study these more rigorously in the future. And these include N-methylpiperazine, diacetyl, and tiglate, and 2-methylbutanoate. And we can all compare this to physostigmine and rivastigmine because of their structural similarities. And here, once again, we have this process of optimizing conditions where we experimented with different bases, catalysts, and solvents. So here are 3 analogs that we ended up putting in our manuscript. And we used pyridine, DMAP, methylene chloride, and toluene to test. And by testing these different conditions, we're able to obtain protocols that would yield give the most significant yield. For example, in our 4-morphine carbonyl chloride analog, we used no base, DMAP, and DCM, which yielded 58.3% product. And once we isolate this product, we try to characterize it using 1H NMR, 13C NMR, and mass spec. And we use this to analyze our product based on the peaks that you see. And you should see a specific, very specific peaks depending on the functional groups of this molecule. So here are some examples. And then we further use computer modeling to perform docking. So we first initialize our molecule structure using a platform like Avogadro, and then we used dense functional theory to optimize our geometric structure, and then finally use this optimized structure to do molecular docking to demonstrate the interaction between our molecule and our target protein. And through DFT and docking, we predicted the binding affinity for each analog that we plan on making. And so here we can see diethyl had the strongest binding affinity versus diethyl, which showed to have the lowest binding affinity of these analogs. And besides computer modeling, we also used colorimetric bioassays to compare the bioactivity of our different analogs. And so acetylcholinesterase catalyzes the hydrolysis of acetylthiocholine that results in this nitrobenzoic acid, which can be used to determine the activity of this enzyme. And we use a plate reader to obtain quantitative data regarding absorbance of our compounds. And we also saw how a higher concentration of our compounds resulted in greater inhibition of our enzyme. As you can see on the right, we use different concentrations for each analog that we're studying. And so our current work revolves around two things. First, we are synthesizing S-rivastigmine and its analogs, so S-dimethyl, S-diethyl, etc., and comparing their biological activity. As reported in literature, R-rivastigmine is more potent than S-rivastigmine, which is its stereoisomer, and we want to see if that relationship holds true for the other stereoisomer. Additionally, we are making our 4-nitrophenyl carbamate analogs using 4-nitrophenyl as our starting material this time to en route to create phenotidine, which is an anticonvulsant. And shown through our colorimetric assays, dimethyl, which has a relatively low steric bulk compared to our other analogs, was the best inhibitor. And so we believe this is due to steric effects. And through our studies with nitrophenyl, which is less massive than our rivastigmine analogs, we want to analyze this relationship. And the way that we can do this is through something called the Taft equation and Taft linear free energy relationships. This equation can shed light on kinetic trends in enzymatic hydrolysis of sterically hindered substrates and the relative sensitivity of each enzyme to steric effects. And so we can compare reaction rate versus steric properties. So here in our future plans, we hope to be able to create this plot and then compare accordingly. So that's our work in chemical neuroscience. Now I'll be talking about another project I'm involved in, which is delving into the mechanistic studies of the long-contested Biginelli multi-component reaction. And a multi-component reaction is just one in which you have 3+ starting materials that are reacting to form a product, in this case, natural. So in the corner here, we have this image of a cell that's in the middle of dividing. And you can see the green spindle fibers here that are attaching to the chromosomes in the middle. And the way that cancer develops is through uncontrolled cell division, or mitosis. And so in the search for anti-cancer therapeutics, scientists at Harvard discovered Menastrol, which is a potent mitosis inhibitor by forming a monoester, and it allosterically inhibits kinesin EG5, which is a motor protein that is important for spindle bipolarity. And so this halts cell division at the stage of metaphase, and especially uncontrolled cell division that characterizes cancer. And so here we see monostrol binding to the kinesin protein and halting its activity. The biomedical potential of monostrol and other 2,4-dihydropyrimidines or DHPMs have extensive applications in medicinal chemistry. They've shown to be good anti-proliferative and antiviral agents, and so we can use them in anti-cancer therapeutics. Moreover, DHPMs have a high binding affinity ligands that can bind to more than one receptor or drug target, which is why they are called privileged scaffolds. And besides menastrol, its derivative LASOM63 will induce apoptotic cell death by inhibiting ecto-5 nucleotidase CD73 activity. So how can we make these anti-cancer agents? Well, to do so, we do this through the Biginelli multi-component reaction, which was discovered in 1893. It involves the acid-catalyzed synthesis of DHPMs with an aldehyde, a beta ketoester, and a urea. What interests us so much about this reaction is that the exact mechanism of this reaction is still being hotly debated after over a century, and there are 3 discussed possible pathways of action. Our goal is to use benchtop NMR to track this reaction and help answer this question once and for all. So we have 3 proposed pathways that have been discussed. The first is the iminium pathway over here, and this is hypothesized to be the kinetically favored pathway, but it involves the condensation of the aldehyde and the urea first to form this iminium intermediate, and then the nucleophilic addition with the ketoester to form the product. The second is the Novanagle pathway, which is the condensation of the aldehyde and the ketoester, and then the resulting carbonium ion intermediate reacts with the urea and cyclizes to form the product. The third pathway is the enamine pathway, involving first the condensation between the urea and the ketoester, leading to a protonated enamine intermediate. And then this all reacts with the aldehyde to form the end product. And using NMR, we try to characterize the 3 intermediates that are possible and help us determine if our molecules go through a certain pathway. So we can compare menastrol and LASOM to their trifluorinated versions, which have not yet been extensively explored. Fluorinated drugs are advantageous to us because of fluorine's electron withdrawing capabilities, as well as improved bioactivity and bioavailability to make more potent therapeutics. Moreover, we can use fluorine NMR to track the kinetics of this reaction this way. Our goal is to synthesize these trifluoro analogs via substitution of the C6 methyl group, the C6 methyl group with a trifluoro methyl group using ethyl 4,4,4-trifluoroacetoacetate as our beta ketoester. And we use different substituted benzaldehydes, which have more or less electron withdrawing properties, to see if that affects mechanistic commitment to a particular pathway. We use ytterbium triflate as our catalyst, acetonitrile as our solvent, and stir on reflux, and we take proton NMR and half-hour intervals to observe the product that's forming. In addition, we can use 19F NMR to track the formation of our trifluorinated compounds as they progress through the reaction and try to determine the rate-limiting step. And we perform this in triplets for each aryl substituent that we have, and analyze the concentration versus time graphs over here. Using this method, we observe an intermediate peak around 77 ppm, which I'll show on the next slides. And this is thought to be the Novanagle adduct, indicating that the reaction goes through the Novanagle pathway. However, our more recent studies with 3 and 4 fluorobenzaldehyde experience cast doubt on this, and for a variety of reasons. So our mechanistic studies so far are inconclusive, but we continue testing with various aryl aldehydes with different 3 and 4 position substitutions, in hopes of determining how this substitution will affect commitment to one of the routes. Over here, in the example, we can see a peak around 77 ppm, which we have yet to characterize this intermediate for. That's why it's unknown. And so that was thought to be the Neuroactive adduct, but we're not completely sure now. So as mentioned, we perform this same optimized protocol for trifluorinated with benzaldehydes that have a variety of different aryl substituents, like aryl halides and hydroxy groups. And they have, in turn, different electron withdrawing capabilities and are thus better or worse leaving groups in nucleophilic substitution. Furthermore, we can use Hammett linear free energy relationships to compare the mechanistic roots of these different substituted benzaldehydes. And this relationship has been used to relate differences in reactivity of substituted aromatic substrates to the electronic effects of these substituents. Lewis Hammett observed a linear correlation between aryl substitution and pKa of benzoic acid. And so we can see here different substituents with different pKa's. And the higher the pKa, the more electron donating the substituent is. However, the more electron withdrawing the substituent is, the lower the pKa. And the more electron withdrawing substituents should better stabilize a particular transition state, making our reaction occur faster. And in doing so, we can see it, and in doing so, this gives insight into how sensitive an addition step is to electronic and resonance effects, depending on the substituted group. And we model this relationship with a Hammett plot. So you can see on the x-axis the Hammett values of sigma rho, which is a proxy for the pKa of the benzoic acid, and the y-axis is the K versus KH seen over here, and this just compares the relative rate of the substituted benzoic material with just a regular benzene. And a nonlinear plot would suggest that compounds within the series don't adopt the same mechanism, but if we do observe a linear correlation, as Hammett did, then that can give us insight into if these are all going through a particular mechanism. So again, these are the three possible mechanisms that our benzaldehydes go through. And for each substituted benzaldehyde, we plan to create concentration over time graphs and concentration versus product graphs and use that slope to make the actual Hammett plot. So over here, our mechanistic studies are still in progress. However, our slope of our products are pretty consistent with the electron withdrawing effects of our functional groups. So for example, the one with nitrobenzaldehyde has the flattest slope in comparison to others that are more electron withdrawing and therefore would have faster reaction rates. Besides all of this, we can do further experiments to gain a mechanistic insight, and that's mainly through our no thiourea and no benzaldehyde experiments. So in these experiments, we just exclude one of the aforementioned reagents, and we can see if the remaining reagents in the same conditions will react and form the suspected intermediate. And if these reactions do not progress at all, then we can gain similar information about the mechanistic root. And so with our no thiourea experiment, we're trying to look into the Novanagle pathway that would form. And then with our no benzaldehyde experiment, we're trying to look into the enamine pathway. So over here, you can see comparison of NMRs of monastrol and trifluorinated on the NMR. You guys can notice similar peaks around until it comes to around here. And why this is when performing the Biginelli with the fluorinated ethyl acetoacetate, before we can form this end product here, we isolate a product that is the hemi-aminal version of it, and it basically just has an additional OH group attached to it, and that results in the additional NMR peaks. And so in trying to synthesize trifluorinated, we have to do an additional dehydration elimination reaction with these conditions under reflux. And after that, we have formed trifluorinated, and then we can use that to conduct biological assays and evaluate their bioactivity, especially in comparison to regular monashal. And with our future bio work, we'll be doing MTT assays on 3 different cell lines. And if you guys are wondering what happened to LASOM63, we have yet to synthesize trifluoro-LSM successfully on a large scale. And so that's definitely in our future directions. But overall, we're still in the process of our mechanistic studies and hope to have solid results very soon. So that is the end of my presentation. Before I end, I would like to acknowledge ASDRP for the resources to conduct my research, Ed for my guidance and mentorship, and my subgroup members and friends. And finally, I want to say, because it's International Women's Day, happy International Women's Day to my women in STEM. So yeah, I'm done. Thank you.
Adrian, that was awesome. Do we have any questions?
I have one. But first, I'd like to say that was an excellent presentation. Thank you very much. And I can tell how much work you put into it. You mentioned at the beginning of your presentation the interchangeable parts idea as being what combinatorial chemistry is about. Is that right? My understanding of combinatorial chemistry is that, from your explanation, is that there are some molecules that have some kind of attachment sites, maybe 3 different attachment sites, and you load them with tons of possible things that could attach to those sites, and then you get a wide spectrum of analogs.
Yeah, so I'll go back to my slide about analog design. Hold on. Sorry.
Oh, no problem.
Okay, so in the analog design, this is an example of the work that we're doing with monashal, right? And you can see how like they have the same basic structure, except for like, for example, a functional group on this very right? And so that's kind of the interchangeable part. And depending on what part we put on, this changes like the functionalities of the resulting molecule, and then we can compare them to see if they're more or less effective.
So it's like you have an original molecule that you're interested in, and so you swap out a certain site that has different functional groups that can attach to it to see if by swapping that functional group, you get a better utility of the molecule.
Yes.
I understand. That's all. Thank you very much.
No problem.